We are here now with Katrin, also from the team from the Medical University in Graz, also part of the MICROBE project, and I'm glad that she also likes to tell a little bit what she's doing and also in the project.
My name is Katrin Panzitt. I'm originally from Graz and I also studied here in Graz. I studied molecular biology and did my PhD, my master's thesis at UCSF in San Francisco. And after that I returned to Graz back and started my PhD with Professor Satlokal. That was in 2001. And did my PhD with him, finished that, and gave birth to 2 children after that. And after the children, we moved to the United States for a couple of years, where I did a postdoc, and we returned from there briefly 10 years ago.
And what is your specialization now in terms of research and science?
Yes, so my specialization has always been the liver. Already during my PhD thesis with Professor Satlokal, we were specialized on the liver and metabolism of the liver, cancer a little bit. That is also, liver metabolism is also the subject where I habilitated on. That was 3 years ago. And we did a lot of sequencing also of human liver tissue and transcriptomics analysis, in general omics analysis. And this is also my part in the project in MICROBE. The sequencing part, recently lots of changes and have happened in the sequencing world, I would say. Nanopore sequencing has entered the stage, and so we are applying it now to different samples. We started with soil and we'll go through different sample types in this project.
Okay. And yeah, that's also already to the question, what are you doing in MICROBE? Mm-hmm. Maybe you can a little bit further elaborate your goals also with your work in the project.
Mm-hmm. So as I said, I'm mainly active in the sequencing part of it. We're closely in contact with the work people from England, and so for now we have performed sequencing, Oxford Nanopore direct sequencing without amplification, which I think nobody else is doing in the project, because some other people are also applying Oxford Nanopore, but not the same type of nanopore sequencing. And so this is finished, and we were the first group to actually upload the data to the Work Package 3 platform they provided. So this should be finished right now, and this is basically our part in the soil, and now we're moving on more towards the skin and throat microbiome. We're at the Medical University of Graz, so we have good access to human samples, and this is our next goal.
We also would like to know a little bit why MICROBE is so important for you or your team, your organization, or especially your group, maybe your research group.
Mm-hmm. So I think our group is also involved in other projects that deal with infectious pathogens, so basically the bad microbes. And so for me it's good to have a project where it's not only about bad guys, bad microbes. So we have a BSL-3 facility down here in the -1 floor, and so this is where the infectious studies are performed. So I'm very happy to not go there all too often and gain more insight into other types of microbes that are not highly infectious.
Sounds also very promising. Maybe you can also tell a little bit what you expect to reach till the end of the project. So we have now 1 year, and we were running 4 years. Mm-hmm. What do you see at the end of the project?
So honestly, I'm really interested in the agglomeration of all the sequencing data from different platforms. This is one of my personal interests. I want to see when we can compare different sequencing platforms and when we can merge the data, how it looks like, whether the optimal combination will be different platforms brought together. So I really hope we can sequence and find the combination of the most bacteria and microbes in soil so far. Yeah, this is that I'm really interested in. And for the development and the future of the project, I think it will be the throat microbiome is of special interest to me, because also their pathogens are residents, frequent residents, especially in spring, where we sometimes catch a cold. And so I think once our ethics approval is through, then it will be very interesting to see also maybe to sample people who fall sick with a cold and then are healthy again, and to compare these sequences and see what is there and what is there under normal conditions. So yes.
We have another question, maybe more personal one. It's what are you doing when not working for MICROBE, or maybe also outside the lab, or with your family maybe?
Yes, we do a lot of things with the family, usually outdoor sports. We like to go hiking, all of us, skiing. We like to go on sometimes even weekend trips, do some sightseeing. That is basically with the family. I personally, I'm a horseback rider. If I have free time, which is not so often the case, I spend every minute with the horse. I have half a horse, so I'm sharing a horse with someone, and yeah, so I like to do jumping and dressage, and now in spring it's very nice to go on long walks with the horse. So yeah, this is where I can regenerate when I'm in the forest, and just with the horse, this relaxes me a lot.
Yeah, it's a nice hobby with another living being. So thanks a lot for your answers, and it was very interesting. And yeah, I hope you will reach your project goals and also all other goals. Thank you.
Thank you very much for the interview.